PhD student for the University of Melbourne and Museum Victoria in Melbourne as well. And today I'm going to talk to you about some of the research I've been doing as part of my PhD project, working on the phylogenomics of the Pulmonate land snails. This is work I've been doing with a number of collaborators as well as my supervisor at as well. Okay, so the group that's traditionally been named Pulmonata is one of the major groups within the gastropods. It incorporates at least 20,000 species. And also includes the majority of the land snails as well as many marine and freshwater taxa. Despite this being such a large group, the systematics of this group has been in flux over the last 100 or so years, with many morphological and molecular studies coming up with very incongruent topologies. This is just one recent example. So this happened, this study was released quite recently in 2010. And this actually showed that the traditional grouping Pulmonata was actually polyphyletic. So it turned out that a lot of traditionally non-pulmonate taxa were actually closely related and within the traditional pulmonate taxa. This includes taxa such as the Staphylococcus sea slugs, the Rhytididae and other sea slugs, and freshwater taxa such as the Glycidoidae and the parasitic Camaenidae. They weren't thought to be pulmonates, but now they've been included in this large monophyletic clade called Pulmonata, and this has been supported in a number of studies, including phylogenomic studies. Okay. So while we've got this new clade called Pulmonata, the evolutionary relationships within this group are still relatively unknown and unresolved, and this has various implications for how we interpret the evolutionary relationships within this group. So I'm particularly interested in the Phylogenetic, which is the major radiation of the land snails. And this is two recent studies, both of which use 2 or 3 mitochondrial genes and at least 1 nuclear gene, but they come up with very dissimilar topologies. And so in this case, the Phylogenetic for the land snails, the sister clade is the Glycidoidae, which are a freshwater lineage, whereas in this case, the sister group to the Phylogenetic for the land snails is a clade including many marine and semi-terrestrial taxa. So one of the questions I want to ask by investigating the Pulmonata is looking into habitat transitions. As I just said, the different phylogenies are suggesting very different transition scenarios for the Pulmonates. Some of the questions I want to try and answer are how many transitions have there been, and did the land snails originate from marine or freshwater ancestors? Okay, so as I said, there's been incongruence and poor resolution in a lot of the recent molecular studies looking at the relationships within Pulmonata. One reason for this is it's a group that's sort of characterized by relatively rapid radiations and morphological convergence. But it's still not clear whether a lack of information on both sides is also leading to this incongruence. So this is where high-throughput sequencing comes in. So the broad aims of my study are to identify a large set of homologous genes with which I can construct a molecular phylogeny of Pulmonata using transcriptome sequences. I then extend this work by using the transcriptome sequences to design targeted enrichment or exon kits with which to investigate specific land snail families in more detail. So if I've got time, I'll tell you a little bit about that target enrichment work as well. Okay, so for the transcriptome study, we've got a data set of 79 transcriptomes, 16 of which were available on NCBI. And so what I tried to do was get at least one representative of all the major superfamilies within this major clade. And we got about 85% of the superfamilies. So just very briefly on the analysis, run Traumatic on the high-throughput sequencing reads to remove adapters and poor quality sequence, do a DNA assembly using Trinity, and then we identified homologous sequences using the Lotti gigantea genome as a reference. I'm not going to go into detail about the orthology determination process we did, but this work has just been published in Molecular Ecology Resources, so go there if you want more detail on that. So we've got this big phylogenomic data set, we obtained 500 single copy orthologous genes. This represents an alignment of 160,000 amino acids approximately, and it's 86% character complete. We took a number of different approaches to partitioning the data set, and most of them came up with exactly the same results. One approach we took, which was a bit different, is clustering exons based on amino acid frequency and variability. And we analysed these in various frameworks using maximum likelihood and Bayesian techniques, as well as we did a dating analysis in MCMC tree. So this is the phylogeny we have for Pulmonate at the moment. Don't worry too much about the detail, but as you can see, we've got 100% bootstrap support for the monophyly of Pulmonate. And we also show 100% bootstrap support for this clade uniting the land snails up here with the marine and semi-terrestrial taxa. So that's come from one of the previous phylogenies that I showed you before, and this is consistent across our analyses. Okay, and so this analysis is still quite preliminary, but I mapped the habitat transitions onto a calibrated phylogeny that I produced in MCMC tree using 10 fossils. And as you can see, this suggests that there's been at least 2 transitions into terrestrial environment, perhaps 3, but there's still a bit of ambiguity as to what's going on here. And yeah, that's what I want to say. Cool. And just for interpreting this data phylogeny as well, there seems to be, it's a bit still a bit uncertain, but it seems like the Phylogenetic originated around 250 to 200 million years ago. So this is all still quite new, but it suggests that maybe there's something going on in that time after the mass extinction event that may have led to this emergence onto land and this big radiation. We also get, you can kind of see a clustering of the nodes at around this time point, and this is broadly covering the Cretaceous. And so these clusters is where most of the families within the land snails originated. So that's something we're going to investigate further as well. Okay, so I think I've got time to talk about the exon capture. So with the transcriptome sequencing, you need fresh RNA, and it's also relatively expensive. So we wanted to work in using exon capture so that we could take advantage of the ethanol preserved specimens that are available in museums. So using this technique, you design specific probes to target a set of genes, where you're targeting the set of 500 genes you developed from the transcriptome sequencing to sequence those 500 genes from genomic DNA. So the first study we did was focusing on the Australian radiation of the Camaenidae. This is a herbivorous land snail that varies in size. And we designed a probe kit based on 2 of the transcriptomes we had, and these 2 transcriptomes, I'll get to that later. So we designed these baits in conjunction with microarray using their MyBaits custom capture kit. And this is in solution hybridization with 120 base RNA probes that can handle mismatch up to 12% sequence divergence. And this probe kit was targeting 2,600 exons. So this is just to demonstrate why we were designing probe kits. This is the pairwise P distance between 2 taxa that cover the Australian radiation of the Rhytididae, the Camaenidae, and Pulmonate as a whole. So that's all of the Phylogenetic as well as those marine and semi-terrestrial taxa. And this is for the 500 genes, so it just shows that across the families, the sequence divergence drops off at about 12%, so it's about perfect for exon capture. Whereas if we were to try and design a probe kit for across Pulmonate, we'd need to include a lot more taxa in our probe kit, and therefore the number of loci we'd have to target would go down. We'd be able to target would go down. So this is just some preliminary results. This is for a test run on 22 taxa, including ethanol preserved specimens up to 35 years old. And on average, we were getting 78% of the reads on target using a double hybridization. 93% of, getting on average 93% of the exons for each species, and an average read coverage depth of 120. And this is just a preliminary phylogeny. Don't worry too much about the details, other than we've got quite good structure for a lot of the major community lineages within Australia. We've now extended the sequencing to several plates, and that's being analysed at the moment. We've also been working on the Australian Rhytididae, so they're a carnivorous clade of land snails that have a good one distribution. We did the probe kit a little bit differently in this case. So we included 6 sets of references for the same 500 genes in one capture kit. And we had 10 transcriptomes of the Rhytididae to work on as a reference. We used 3 species, the sequences from 3 species, but we also used 3 sets of ancestral state reconstructions to create probes from. So we did this in an effort to capture sequences from lineages that were quite deep and divergent, but we didn't have transcriptomes to represent that. Cool. And so this analysis is also very preliminary, only a week or 2 ago. And the really cool thing about this analysis, this is for about 140 specimens of the Rhytididae across South Africa, New Zealand, and Australia. We get really lovely structure within a lot of the major lineages and clades, but we get this really big broad backbone of very poor resolution. But the cool thing about this is because we've used 500 exons in this case, it's lending evidence towards the fact that this might actually be real. And this is a South African lineage, and there's South African, New Zealand, and Australian lineages in here. And given some previous analyses, it's what we want to try and work out is where the Gondwana breakup is relative to these clades, and whether this is the Gondwana breakup or this one, and something strange went on here. So we're going to analyse that better. Cool. And so in the future, we'll be doing more exon capture on various families and clades, and we've got collaborators working on the Helicoidea and the scenic gastropods. But the really cool thing is that now that we've got all this transcriptome sequencing, anyone who's interested in any of these families of land snails or even some of the marine and freshwater taxa can take this data and design their own exon capture probes for these genes and work on their group in detail. I'd just like to thank my supervisors and collaborators and my funding sources.
Plenty of time for questions. So are you putting exons, like multiple exons from the same gene together in your alignment, or are you building just single exon alignments?
For the transcriptome or for the exon capture?
For your capture, when you start building
Three separate alignments for each other.
Okay.
And so you actually get a lot of flapping region as well. We've been de novo assembling the capture region.
Yeah, I guess I was wondering about the utility of using a closed genome to identify smaller genes with like shorter introns that you could span exon to exon and actually a full length gene for gene capture.
The trouble is there's not much genomic resources, so we actually found that we use the exon boundaries of Rhytididae, which is like really, really divergent, and we found that a third of the genes actually contained more exons than we thought from the Rhytididae. So it was actually really cool to look at the genome evolution.
You said you used ancestral state sequences to construct exon probes. Did that improve the data set a lot, or like how much improvement?
It's a bit hard to compare because we don't have the alternative where we didn't use those, but we were able to capture sequences from the deep lineages that we didn't have transcriptomes for, so it didn't hurt, is what I'm trying to say here. Yeah.
Why would the breakup of not give you short internets?
Not entirely sure, but there's various. The only thing I can think of is that there was one, or like a limited number of species, and then when this breakup was created, it led to diversification of habitat, and so these radiations occurred quite quickly.